Hello anatomy students. In these next two videos, we're going to study the neuron. The neuron is the functional cell of our nervous system, which functions to collect stimuli from the environment, process it, and stimulate our body to respond appropriately to those stimuli. And so we need to know how neurons work in order to truly understand how all of this occurs. So I've broken up this topic into two chunks. Our first chunk is talking about the electrical nature of neurons. What are the characteristics that these cells have that are going to allow them to function within our nervous system? And then the second part is really digging into an action potential and what does the trace of an action potential actually mean, and what is actually happening at the cellular level to allow all of that to happen. So that's going to be in part two. This is part one. We're going to focus just on the general characteristics of neurons. What you're seeing in the picture here on my first slide is a picture of something called a voltmeter. A voltmeter is an instrument that will measure the potential, the membrane potential of a cell. And what we have off to the side are two electrodes, one that will be placed outside of the cell in the extracellular fluid and one that will be placed inside of the cell in the intracellular fluid. And it's going to measure the difference in charge between the outside and the inside of the cell. And what we get is this red line, or our trace, and that's going to provide us with some numeric data, and we'll be able to follow along and see, oh, why is this line going up? Why is the membrane potential kind of narrowing? And why is it getting bigger? So those are some questions that you're going to be able to answer after watching these two videos. So I just wanted to give you a brief introduction on this voltmeter because you're going to be seeing it a couple of times during this presentation. So after listening to this, you should be able to explain what membrane potentials are and what resting potentials are and how they are maintained. And number two, describe the types of channels that are found in the cell membranes of neurons, because those are the things that are going to maintain the excitability of these cells, which is really important. You know, if we put our hand on a hot stove, we want to be able to react right away. We don't want to have to wait a couple of beats, during which time we could damage our tissues before we respond. We want to be able to respond right away, and the channels are going to allow us to do that. Okay, so a couple of things to keep in mind as we begin. First of all, neurons conduct nerve impulses. We're going to talk more about those as action potentials in the second video. Neurons are excitable and conductive, which basically means that they can be stimulated to open channels and to change the ions that are found inside and outside of their cell membrane. But they're also conductive, and these signals, these changes, can spread throughout their cell membrane if they are strong enough. The two ions that we're going to talk about the most are sodium and potassium. So sodium is Na with a +1 charge, and potassium is K with a +1 charge. So I want you to notice that both of them are positively charged. Finally, ions are going to try to achieve equilibrium, which means that they're going to move so that they have equal amounts of themselves on either side of the cell membrane. But they're going to be foiled at some point because of the channels that exist in the cell membrane of the neuron. There are also some other factors in the cell membrane too that are going to prevent this equilibrium from happening. We don't want this equilibrium to happen, you see. We want there to be a gradient so that changes can happen very, very quickly, and therefore we can respond to stimuli very, very quickly. All right, so let's get going. First, let's talk about membrane potentials. A membrane potential is simply the measure of charge outside of the cell and inside of the cell, and then you subtract them in order to see what their difference is. So a membrane potential is just the difference of charge between the intracellular fluid, the fluid on the inside of the cell, and the extracellular fluid, or the fluid that's outside of the cell. We call it a potential because it is a kind of potential energy. So it is it is a kind of energy, so that's why it's called a potential. But a membrane potential is simply a difference in charge. So if you take a look at our picture down here, you can see that one of the electrodes, like I mentioned before, is measuring the potential outside of the cell membrane, while the second one is measuring the charge inside of the cell membrane. And then our voltmeter is going to subtract them, and in this example, we are getting a difference, an electrical difference of negative 70 millivolts. So here's what that means. The negative sign means that the charge of the intracellular fluid, the charge inside the cell, is negative, and the 70 is the difference in charge between the outside and the inside. So whenever you look at the charge of a membrane potential for neurons, we are really looking at that sign is going to tell you what's going on inside of the cell. So the sign, be it negative or positive, in this case it's negative, tells you what the charge is on the inside of the cell, not on the outside. And so that's what the voltmeter does. And then of course that red line is showing us like the trace. So with this measurement, we are getting a constant reading of negative 70 millivolts. Actually, negative 70 millivolts is something that we call the RMP, or the resting membrane potential. So when a neuron is not in an excitable state, when it is just resting, we have a resting membrane potential of negative 70 millivolts. So the difference in charge between the inside and the outside is 70, and the negative sign tells us that our intracellular fluid is negatively charged. So what's happening here is that we have some ionic imbalance, obviously. Otherwise, we wouldn't have a difference, right? If we had equal amounts of all kinds of ions on each side of the membrane, then that red line on our voltmeter would have been at 0, reading no difference in charge, right? But we do have a difference in charge, and the inside of our cell is negative. Here's why. The inside of our cell has chloride, which is a negatively charged ion, and we also have other molecules, proteins, that are labeled here in our picture as anionic proteins. So maybe you remember from chemistry that positively charged ions are called cations. Well, negatively charged ions are called anions. And so anionic proteins just mean that they carry a negative charge too. There's very little chlorine that is in the cell exterior, like right next to the cell membrane. And so you've got more negative charge inside of the cell than you do outside of the cell. And that's what's giving us the negative in the negative 70 reading. So ionic imbalance is also maintained by different kinds of channels that are found in the cell membrane. The first type of channel that I want to talk about is a gated channel. The sodium channels are gated channels, and you can see here that we have a closed sodium channel here. You can see that the doors are shut on both of these sodium gated channels, and so all of our pink sodium ions are found outside of the cell. At least most of them are. You might have a few that are trapped inside, but they can't get out and they can't get in because at rest, these gated sodium ion channels are closed. The other thing to notice about this picture is that our purple potassium ions are found mostly on the inside of the cell. And you'll see that we have a closed potassium channel, so that's a gated channel. But we do have some leak channels here that are embedded within the cell membrane. Leak channels are going to allow a limited number of potassium ions to constantly flow out of the cell. And so here you can see that we do have some potassium ions that have leaked out of the cell, and they're in the extracellular fluid, and that is also going to contribute to the negative charge of the intracellular fluid inside of the cell. So not only do you have chloride and anionic proteins, but you also have some positive charge always leaving the cell, which is going to make that intracellular fluid more negatively charged than the extracellular fluid. Oh, and one more thing about that. I just saw that last bullet here, and I wanted to make sure that you knew that. Notice that in this picture, we don't have any channels for anions, and that's because they can't readily move in and out of the cell. Okay, another channel that we have embedded within the membrane of our neuron is called the sodium-potassium pump. And this sodium-potassium pump is also responsible for maintaining our resting membrane potential, or our RMP, at about a negative 70 millivolts. So the sodium-potassium pump is going to use active transport, which means that it requires ATP, which you can see here inside of our picture. So here's our ATP molecule. And when our pump is activated with ATP, it is going to force 3 sodium ions out of the cell and pull 2 potassium ions back into the cell. So notice that we have a 3-to-2 ratio. We have an imbalance, right? The sodium-potassium pump is going to help maintain this electrical imbalance, this resting potential at negative 70, because it's going to purposefully create this ionic imbalance. It's pushing more positive charge out of the cell than it is drawing positive charge into the cell. And again, that's to help this neuron maintain an excitability potential. What this means is that, if you think about maybe a lake or a stream or a river that is dammed, right? All of the water is going to collect behind the dam. But when the gates of the dam open, the water rushes in. That's exactly what's happening here. By making this imbalance in charge, by pushing more positive ions outside of the cell than there are inside of the cell, when these gates open, when these gated ion channels open, you're going to get a rush of ions coming in. And it happens so fast. I mean, it's going to take me like several minutes to explain this, but in our bodies, it's happening super duper fast. And the reason why we're able to respond almost instantaneously to stimuli is because of this ionic imbalance that exists on each side of the plasma membrane. So I wanted to talk a little bit about local potentials, because not every stimulus is going to be propagated through a neuron. That would be like too much stuff going on, and we just would not be able to handle it. So we're going to have multiple local potentials that are going, to occur, but it doesn't mean that they're all going to cause a response, or we'd be responding all the time. So we have to be, our bodies have to be selective. Our neurons have to be selective. So local potentials are fluctuations that are occurring either above or below resting, so above or below negative 70 millivolts, in a very tiny area of the neuron's plasma membrane. And so the way that this happens is we've got excitation that occurs. We have our stimulus-gated sodium channels. So those are the sodium channels that I showed you. Let me just go back to this picture. The sodium channels that we have here shown in green, these are stimulus-gated or ligand-gated sodium channels. And they are going to open when either a molecule binds to them, or when there's a certain membrane potential around them that is reached. So these are called stimulus-gated. They're only going to open up and allow sodium to flow in when they have been stimulated, hence the name, to do so. You have multiple stimulus-gated sodium channels in the input zone of the neuron, so where the dendrites are and where the dendrites meet up with the cell body, because that's where the neuron is going to accept or receive stimuli from other neurons that live around it. So when the sodium channels open, sodium is going to flow into the cell. So now you've got an influx of positive charge coming into the cell. And that is going to make that resting potential, which was negative 70, become more positive, go a little bit more towards zero. This is called depolarization. You see, if you think back to your chemistry class, right? Think about molecules that are polar molecules like water, right? We call them polar because one side of the molecule is positively charged and one side is negatively charged. That's exactly what we have in a resting neuron. The outside of the cell is positive, the inside of the cell is negative. So at rest, the neuron is polarized, the cell membrane is polarized. But when you open up the sodium gated channels and sodium ions flow into the cell, making the inside of the cell more positive, well, you're starting to equalize the charge on each side of the membrane. You're bringing them closer together. And in our drawing here, this little hill that we have coming up, it's going more towards zero on our picture. This is called depolarization. We are undoing the separation of charge by allowing sodium to come into the cell. We are driving that charge inside of the cell to become more positive, to go more towards zero. So that's something called depolarization. So when the membrane potential rises towards zero above negative 70, that's called depolarization. At rest, the cell membrane is polarized. All right, so remember that local potentials are simply fluctuations above and below resting membrane potential, and we just talked about depolarization, but not all local potentials cause depolarization. Some of them are inhibitory, which means that they're going to stop the propagation of action potentials from happening throughout the entire cell. If a stimulus is inhibitory, it's not going to stimulate sodium channels to open. No, it's going to stimulate potassium channels to open. So let's go back to that picture again. So notice that we have more potassium inside of the cell than out, but if we have an inhibitory signal, it's going to cause our red channel to open. Potassium is going to flow out, which means that the inside of the cell is going to become more negative. Why? Because it's losing more positive charge. So when that happens, that's going to cause hyperpolarization. So when you have a fluctuation below resting potential, below negative 70, that's something that's called hyperpolarization. So you have to think about what's going on at the subatomic level, really, and you've got to think about like what is actually happening with the sodium and the potassium, like what's flowing in, what's flowing out, and also you have to remember that the inside of the cell is more negative than the outside, and that should help you remember what's going on with depolarization and hyperpolarization. Okay, so remember that what I was just talking about were local potentials, right? You're going to have local potentials that are happening all over the cell. We're only going to get an action potential that's going to be carried through the axon to another cell if the sum of all of these local potentials reaches a certain measurement called threshold, which we'll talk about in our next video. But local potentials are different from action potentials. Local potentials are little tiny ones that could add up to create an action potential. So local potentials are isolated. They're going to be found within a particular area. They don't spread down the whole axon, not yet. And they can be very large and contribute to an action potential or not. Local potentials are not considered all or none events. Action potentials are, but local potentials are not. And they are going to exhibit decremental conduction, which means that as they spread, they're going to become weaker. And so they're only going to contribute to an actual action potential if there are others similar to them that are going to like summate or add together in order to reach that threshold number. So local potentials are going to add together to create an action potential. All right, so let's see how much you understand from our little talk here. Number 1, define a membrane potential. I'll give you a 5 count. Well, the membrane potential is simply a measurement of the difference of charge from the outside of the cell to the inside of the cell. And so there's your answer. Number 2, where is there an excess of positive charge around the neuron? If you said that there's more positive charge outside of the cell membrane in the extracellular fluid, then you are correct. Number 3, what does the negative sign indicate in negative 70 millivolts? If you said that that shows us the charge of the intracellular fluid or the charge inside of the cell, then you were correct. Number 4, which type of channel opens in response to a signal molecule that binds to a receptor? If you said stimulus-gated or ligand-gated channels, then you are correct. Last question. Movement of a membrane potential toward 0, so away from resting, which is negative 70 millivolts, and towards 0, what do we call that? That is depolarization. Okay, anatomy students, thanks for watching part 1. You should move on to part 2. Hopefully this will clarify some things for you as we talk more about nerve signaling.